Good afternoon, everyone, and welcome to the EAStalk webinar hosted by the European Aquaculture Society. My name is Anna Viskovic, and I'm the Project and Program Manager here at the EAS. It is a pleasure to have you all with us today. EAStalk is a series of webinars designed to showcase the multidisciplinary nature of aquaculture. Today, we are focusing on animal welfare, a topic that is becoming increasingly important across both aquaculture and livestock sectors. Reducing our reliance on veterinary drugs is a key part of improving animal health and well-being. It also helps reduce the presence of pharmaceutical residues in human food chain. And one promising approach to address this challenge is the use of electrochemical sensors, which enable real-time and continuous monitoring of biological and environmental conditions. The work we will hear about today is being developed as part of the Ignition Horizon Europe project. It is my pleasure to introduce today's speaker, Dr. Sofia Teixeira, Senior Researcher at Tyndall National Institute in Ireland. Dr. Teixeira received her PhD from Swansea University in 2014, specializing in graphite biosensors. After completing her doctorate, she worked as a National Research Fellow at Swansea, contributing to development of epigenetic biomarker tools for ovarian cancer diagnostics. Her current research focuses on electrochemical biosensors tailored for a broad range of applications. Her expertise includes sensor fabrication, electrochemically enhanced diagnostics, and sample processing techniques. In today's presentation, she will share her recent work on designing a new generation of electrochemical sensors aimed at improving animal welfare. Dr. Teixeira, welcome and thank you for joining us. Before we begin, just a few quick reminders for everyone. Today's session will last for approximately 45 minutes. Dr. Teixeira will give a 30 minutes presentation, which will be followed by a 15 minutes live Q&A. So you can submit questions at any time using the Q&A box or a chat. So feel free to type any questions that you might be having. And please note that this webinar is being recorded and the recording will be available for 1 month on the EAS YouTube channel, after which it will be available exclusively to EAS members in the EAStalk webinars. The presentation slides will also be available for the EAS member. Thank you very much, Sofia. You're free to go and start with the presentation. And thank you again for joining us.
Thank you so much, Anna, for your introduction and your kind words, and welcome everyone to this webinar. So today I will talk about how an electrochemical sensor can be as a platform to improve animal welfare. So, and for this, I'm going to give a little background on Tyndall, so the institute I worked with. After a little about electrochemical sensors and what they are, and how they can be tailored for aquaculture. And after, I'm going to show you a little of specific case studies that we've been developing in the projects in Ignition. And after, I will show you how we moved from commercial sensors and you went through facilities of printing these ones and tailor them for a different approach. So a little about Tyndall. So Tyndall is a leading European research center in information communication technology, materials, device, and systems. We are mainly specialized in electronics and photonics. We are the flagship for UCC. So UCC is a University College Cork. We are a home for approximately 600 people from around 57 nationalities. This include around 108 PhD or master students. We have as well top facilities that receive 250 million in infrastructure investment, and we generate around 47 million of annual income. 80% of our funding is from competitive fundings. but we work very close with industry, and what Tyndall have is, apart from the common of European and national projects, working with the company gives us a different impact and help us to focus in a more delivered to human and economic impact as well, and improve our research excellence. So talking a little about Tyndall, we will move away from what actually are electrochemical sensors. So I'm not sure if the majority of people here is familiarized what is electrochemical sensors, but usually sensors are made in a more close process using clean room fabrication models, as the ones that you can see here on my left, and they are more rigid platforms. You can miniaturize them to different scales, but they don't address some challenges that we need, that is the integration in flexible and stretchable substrates that can be conformal with life science applications, even if it's for human, aquaculture, environment. So for addressing this challenge of the flexibility, having biocompatible materials, real-time, minimum invasive, and allow continuous monitoring, we develop these flexible materials. And these flexible and structural platforms can be 2D and 3D based. So depending what is our goal and our interest, the platforms can be in a 3D. So I'm going to give a more specific example. So we basically can have, so this is what we have here. We can have our electronics, and this is the path that comes on the top, and after you can have 2D sensors or 3D sensors. And this can be made from or microneedles or 2D substrates from different base materials. We can print them directly onto balloons or catheters, or we can print it out from them and after attach them to these balloons. You can have the freedom to do different types of sensors in the same electrode. That means can allow a multiplex detection, and as well, not only for one type of biomarker, but having different biomarker, if it's a chemical, a biochemical, or an enzymatic biomarker in the same sensor. So that means with the same fluid, you can have more than one target at that time. The microfluids, the microneedles allowing to have the contact with the skin, so it's non-invasive, but at the same time allow us to measure biomarkers from interstitial fluid. So that means that we don't need to have a drop of blood or an invasive technique to actually measure the same biomarkers that we have in the blood, and we can use the microneedles. And in the end, we can actually integrate everything as different prototypes to have in the field and to test it in different places on the field, if it is in aquaculture or in hospital or at GP level, and we have the freedom to take them with us. So I'm going to start giving you some examples of different sensors that are already been developed. Well, it's just a brief overview. It's not all here, but it's the most important, I guess. So we have biomarkers that goes from infertilization in vitro to infection, to gynecologic cancers, to environment, to aquaculture, to epigenetics. So it's a range of different types of biomarkers for different applications, but as well using different types of materials. So my expertise goes from not just only to modified the sensors for the target of interest, but as well I can do the fabrication of this type of sensors. So all the pictures that you see on the background was developed apart from this one here, because is the commercial ones, they were all developed by me in a clean room. So this is more on the immunosense area. After we have as well in the area of chemical and enzymatic sensors. So for carry on work on the chemical enzymatic, the majority of the work developed for dissolved oxygen and glucose was for adjust and adapt this in bioreactors. So what we wanted is to monitor in different points of the bioreactors how the dissolved oxygen was changing with the changing of the cells, and as well how the glucose was being modified in the media of that. So this means was everything non-invasive. You only have to put the sensors into these bioreactors, and from there we could have real-time monitoring that goes to an app of a phone or a laptop. We had as well some sensors that we developed for lactate, but for these ones we use on the same system for bioreactors, but as well in parallel for point of care devices using different platforms. In this case, actually, we use graphene platforms. And we can measure different types of pHs sensors. These including on the reactors or including with a multiplex sensor to have that as your background for always understand what is the pH of the system, and if that pH change, how our target can change. So understanding the effect of the pH in this type of sensors is very important to monitor the day by day for a patient, for example. Or even for if we have a tank for fish, it's important to understand the pH and how they change. So in the area of microneedles, so I start collaborating with another PI in the group here in this area because they fabricated the microneedles, and I modified the microneedles for this. And in this case, we have more, the developing of them was more in the area of ions. So they were interested in do a patch that we can put in our skin and monitor for pH, sodium, potassium, and calcium. All of this has been or published or submitted, and we always try to use mimicking solutions to mimicking the human body to understand if that could work in a real environment. And as well, we developed last year a project for a heart failure cardiac biomarker, and this is a company project. So I show this one here because to show you that the company is coming to us. They want that we develop a specific biomarker sensor for them if using microneedles or using any type of sensors. And we try to develop and mimicking what we need for they have a point of care device in the end. And after they can do whatever they want. They can manufacturing, they can sell it. That belongs to them. So when I came to Tyndall, they already have some systems that was, for example, a capsule that goes into bioreactors, was a probe that could go, and here was just a simple example. This is all what they have developed, but as well, they already had a project that allow us to monitoring welfare for aquaculture. That was a FP7 project, and the idea was they have like robotic fish, and in this robotic fish you have different types of controllers from different types of systems that was needed for they understand, but a big part was having here chemical sensors. So they have sensors from pH, potassium, calcium, all the biomarkers of interest for measure inside the water. And actually this was tested in high water sea. And the project was a success. All this part was developed for different partners. This was a European project, but Tyndall developed all the sensors and they incorporated this and they went to test it in the sea. So they already had a culture of trying to work in welfare for aquaculture. So for me, follow up on this was very easy. So we try to understand how can we incorporate these non-invasive sensors into a new platform. So the idea is we identify what specific biomarkers we want, and we don't want that they be invasive, so it's from water or even from the mucus, because it's the easiest and not the blood. And after we identify this, or we do it ourselves, or we buy it commercially, and we can modify these sensors for the specific biomarker of interest. And this is a potentiostat that you can take with you anywhere in the field and add this sensor here. And after, we have an app that allow you to control this system. So this is basically having, everyone knows the glucose sensors, that it's portable and can go with you elsewhere. We can do the same, having our own sensors and potentiostats. So what these biomarkers can be used for. So we can use for different types of things. One is for diagnosis and another one is for screening. For diagnosis is trying to understand the sensitivity, specificity of a certain disease, if it's animal or non-animal. And from here, we can be a prognosed for the outcome and do a treatment. If you want to do a screening, here will be, they are high specific. They will minimize the false positives or negatives. They can be easily detected without having very invasive procedures, and the cost. So when we produce a certain amounts of these sensors, the cost will be decreasing substantially. So what type of samples can we use? Really, we can use any type of samples. So from blood, from mucus, from tissue samples, from water, from any other fluids. If we are talking about humans, you can use ISF, you can use sperm, you can use pus. So all of these are biomarkers that you can use in the sensor, and that will allow you to have an answer for the specific biomarkers that you are interested in. Aquaculture have a broad range of different applications, can understand the stress of the animals, the immunofunctions, some changes on the metabolism, on the behavior, and understand how they change even with certain conditions, how the reproductive capacity can change, and as well to understand the change on growth. So we can have a different range of applications on this field without being invasive techniques, and allowing to understand the welfare of a fish. So now I'm actually going to give you some of examples that we already did in our project Ignition. So I'm going to give a little of background. So first, I'm going to show you very briefly how a sensor is developed. So from having a sensor that has three different important things. For electrode, that is our working electrode, our counter, our reference, everything happen in this working electrode. So this working electrode is where the work of interest will happen. So in this case, we had a graphene electrode, but for this, we need to actually modify this to be able to attach an antibody that can match to the surface and after to the biomarker of interest. So we found a linker that allow this attachment of the antibody. So when we have this and we attach the antibody, in this case, that antibody will detect modifications at level of cortisol, because this was a sensor detect for cortisol. So sometimes we will have some holes or some parts of the sensor that are not completely modified or attached with antibody. So to make sure that is selective enough and that we don't have interference from anything, we blocked that surface first to make sure that everything is covered and only the signal will come from the antibody. So in that moment, we are incubated with the cortisol. so the target of interest. And when we incubate this with a cortisol, we can detect, and we're going to have these changes as electrochemical change. And this change will happen all as these peaks will change. And each peak is correlated with a different concentration of the target of interest. And from there, we can take the limit of detection, we can take the sensitivity of the system. So one of the examples is here. So with these sensors, we detect cortisol, and we could actually have a limit of detection of 5 femtograms. So this was tested in water. But after, to understand this was correlated with the values that we get from so we sacrifice the fish in the end, and we try to understand the difference between the two controls that we have. One was enriched and another was poor. And from this, we did a physiological and behavioral relationship between cortisol levels and the different environments and the body of the animal, and as well with the basal metabolic rate. And this was the most important for us, because show us that the metabolic rate increased significantly with the cortisol levels, but this was not affected at all by the parental enriched environment. So we could see that actually the levels of cortisol that we were measuring from the water was actually correlated with the metabolic levels that they had detected after the system. So another test that we did was in collaboration with CIMAR. So, and here we try to detect cortisol in water, and was for understand chronic stress in zebrafish. So what they did was they individually applied one of 7 different stressors twice a day for 14 days. And after they euthanized the animal to collect the samples. So in addition to this, they measure cortisol in the trunk water and skin mucus, and they try to measure the oxidative stress markers in the brain and liver to actually confirm the chronic stress. So with this, they found that the research shows that only water cortisol actually respond to stress protocols, and the oxidative stress and cholinergic alterations was found at a brain level. So they could conclude that mucus was only reliable to measure acute stress, but water could be reliable for a biomarker for chronic stress. So this change in the oxidative stress markers in the brain confirm this response, and the lack of changes in the liver that they found was maybe due to the high antioxidant capacity comparing to the brain. But we could use actually the water as a chronic stress biomarker. This is what we found from this work. So another work that was developed in Ignition, but with the University of Cadiz, was to detect as well cortisol, but from extracted plasma and mucus. So we had two groups, one control and one stress. So the control group was left undisturbed, so we never touched in that one, and the stress group was subject to air exposure. And we had from 10, 30, 60, and 24 hours, we allowed this fish be under stress, and we measure from plasma and mucus the results. And we actually could see that from both the plasma and the mucus, we could have a very good differentiation how the cortisol could be detected with our sensor, and that was correlated what they could see in the literature and in their facilities from the ELISA tests they did. So the sensor was detecting much lower levels than what they can with their system, but this was in agreement what literature says that was for the fish cortisol levels in plasma and in mucus. So from this, we decide to move forward, and we actually start doing our own flexible sensors. That flexible sensors allow us to be tailored to be as a skin patch, for example, or as a floating egg. So what that means, so we can develop the sensors. The sensors will be modified with the linkers of interest to antibodies and blocked for all the specific biomarkers. But you can have more than one of these ones, and even in the same electrode, more than one biomarker, but you can use it as a patch that can go on the skin of the fish. We can have on the electronics as well a GPS attached, so you know what is the fish that have this, or we can put in tanks, and the sensor will be inside the egg, so it's like a small shape egg. When you have the electronics on the top, the middle you have your sensors, and on the bottom we have weighting, a weight heavy or non as heavy, depending where we want that be disposed on the tank, and at the same time that will allow us to go to a continuous monitoring to a platform or a laptop or a phone. This allows continuous monitoring and multiple sensors, and they can stay up to 30 days at the moment. So the idea is you have this sensor, you incorporate it there, and you can measure a lot of things. So at the moment, we are measuring stress and cytokines using this. We are moving to microRNAs, and this is measured in water or mucus, but everything needs to be scratched from the fish to don't have introducing more stress or a different type of behavior on the fish. So how we do that? So basically, this I'm going to show you, the video is just showing a small example of one that you can print, but we have a machine that allows us to print in high volume, and we can use different types of materials as the baseline. You can use different types of inks from non-compatible to biocompatible. So it's allowing you to have a freedom to actually play with that as you want and the size and shape that you want. So with this in mind, we start for developing some sensors for cortisol. And we have the same conditions that we did in the previous ones, sensors, and we tried to understand if we could have a similar signal or a better signal. And in this case, we actually could go to similar levels that we had in the commercial available sensors, which means that our sensors were working as we expected. So from this, we try to understand if we could incorporate it more than one sensor in the same. So we developed a different sensor in parallel for interleukin. And when we had these two in parallel, the idea was, okay, can we actually develop the two in the same platform? And all the range of every sensor developed here are the range that are clinical relevant ranges, if it's for animals or for humans, but everything needs to be clinical relevant. So the sensors are always developed for that. So having this in mind, we put the both together. So in the same sensor, we modified the surface, and after we change the antibodies that we were having for the cortisol and IL-6, and with the same sample, we try to measure and to understand if this was working for a dual sensor that we could deploy anywhere in a tank or in a patch. And yes, we could. And we had very good limits of detection. So we had 0.023 picograms for cortisol and 0.0016 femtograms for IL-6. So this was a very good preliminary result. So we try to understand if actually we add interference species to these sensors, what's going to happen? And we tried IL-10 for IL-6 and testosterone for cortisol. And we realized that actually the interference was less than 5%. So the significant impact interference of this species on the sensor was not as significant. So the sensor was working as a dual sensor without interferences. So at the same time, I want to show you that not just for this type of sensors, but we could use this for detect, for example, hydrogen peroxide from the tanks. So hydrogen peroxide, you better than me know how is important in aquaculture as a disinfectant in holding tanks, as the losing agent for salmon farming, and high concentrations can be dangerous to fish welfare. So monitoring hydrogen peroxide is actually of a great importance. So what we did was we developed a sensor that it's chemically modified. So for this, we don't need antibodies. We just modify chemically, goes inside the egg. You can add in your tank. You can continuous monitoring. You can have more than one target. So if you want pH and hydrogen peroxide, or if you want hydrogen peroxide and cortisol, or iron peroxide and cytokines, you can do it. And you can stay with these sensors there for approximately 30 days, and you can measure it from water without having non-invasive techniques. So for this, we use a specific detection that was nanoporous gold. And you can clearly see from when we actually had our sensor modified with nanoporous gold and without modification. So everything is happening around -0.1. So we clearly can see the differentiation of the different types of concentrations when we modified, but we are not able to see at all when we don't modified. So with this in mind, we actually did a further comparison to the modified and unmodified for a calibration plot, and we try to understand what we could achieve. And we actually could achieve a very good linear of detection for hydrogen peroxide in the environments, but when we actually don't have anything, it's almost linear, so we couldn't. So that was an important part to understand, that we need to modify the surface for having the concentrations that were relevant to see in tanks from fish tanks, that we needed the modifications for these sensors. So as I hope I show you that a different variety of different possibilities that we can use different types of sensors for different types of applications. So as a take home message, I hope that I show you that electrochemical sensors can be helpful and useful and can be tailored for a range of different aquaculture applications. You can use from stress to cytokines to microRNAs to hydrogen peroxide. So the possibilities are really massive depending what is your target of interest. As well, I show you that for cortisol, IL-6 and hydrogen peroxide already was developed, modified, and we have that already in place. We show in-field detections in CMAR and in CADIS for the detection of cortisol. So I think with all of this, I hope I'm on time. I want to say thank you first to my team to being here and supporting all the work, to all the ignition partners that makes this work actually helpful, because a lot of things I depend on them, to Ignition and all the research funding agencies that makes this work possible, and to all of you for your time, and I'm open for questions if you have it.
Thank you very much, Dr. Teixeira. It was a fascinating presentation, and yeah, I was really looking forward to learn more a bit about the biosensors and the work that you are doing. I think that the work that you shared today was highlighting how cutting edge sensor technology, which is combined with the system design, can be a practical and real-time solution for animal monitoring in aquaculture. And yeah, these developments are definitely supporting not only animal welfare, but also hold great promise for reducing the drug use and improving overall system sustainability in the future. I would like to start with the Q&A right now. So feel free to ask any questions that you might be having. Now is the time to ask Sophia. You also have Sophia's email here, so make sure that you note it somewhere and to get in contact with Dr. Teixeira. So the first question we are having is from Manuel Gesto Rodriguez. For cortisol sensing, you stated an LOT which is less than 1 pH per milliliter. How long should be the contract of the sensor with a sample, for example, water, to attain such sensitivity?
So at this moment, every sample in contact is around 15 minutes. And after 15 minutes, we can have this type of signal.
Thank you. The next question that we got is from Pablo Cabo Valcare. The sensor can be reused. What is the price, and how many sensors by tank do you need to be reliable?
So it depends on the size of the tank. So they can be reusable if they are chemical sensors. If they are immunosensor based, at the moment they cannot, so they will be disposed after. But for we have a more continuous monitoring, what we do in the patch, we allow to have a different arrays of different sensors to be able to be activate only in the time that we want the measurement. So sometimes continuous doesn't mean we want a signal every millisecond, but maybe every 1 hour. So we can have this as the backup. So we can, on our electronic system, we can actually say to the sensor that now we're going to measure. So they have like a layer that protect them before. And only when I say to the system that to read, we have that. In terms of costs, I really don't know. I don't have an idea. This can go to lower than, 10 euros per sensor, but depends the amount of sensors that we actually produce. So if it's fabricating of high volume, probably this cost go much lower. But at the moment, that is more or less where we are at with costs. I'm not sure if I reply all the questions.
I think you did so far, but in case Pablo has some other question to ask, feel free to write it down. The next one we got is from Marcio Moreira. These sensors were already tested in earthen pond cultures, a very enriched environment, and if yes, with the good results.
Not these specific ones, not in earthen pond culture. So everything was for this ferret, for the enrichment environments was in labs. So we actually produced it, the rich and the poor environment. So we never tried in pond cultures at the moment, no.
Thank you. Can you please tell us as well, what is the actual size of the patch and the floating egg?
So the size of the floating egg is around 7 by 4 at the moment. And the size of the sensor really can be depending on what we want. So can go for a few millimeters to bigger, depend how many sensors we want. So that will depends on what actually, how many parameters or how many sensors we want in the same sensor. So we can do as big or as small as we want, depend what is our target.
Thank you. So I also want to know how do you apply these patches? And like, for example, do you use the water bath for the fish, or is there any other method to apply the patches?
So at the moment, what we will do is, so you will have the fish, of course, and we need to have a surface where you actually do a clean for the patch, for the glue goes, but it's waterproofed. And when you put it there, you only have to take in 30 days.
And do the patches leave any marks on the skin?
Not in humans, so I guess not in fish.
That's a great news to know, and it's quite important for aquaculture as well. The next question that we got is from Pablo again. There is tested only in salmonids. So was it only tested in salmonids?
So at the moment, this was just tested in, well, I'm not sure what means with salmonids. Maybe this is not my area, so I'm not sure what that means. So this was tested in water tanks in different places and was tested in blood and mucus from fish. So I'm not sure if I replied to the question because I'm not sure what that means with salmonids.
I think he's asking which fish did you use?
Oh, yeah, okay, thank you. We use a range of different ones. So we had some sea bass, sea bream, and salmon.
Perfect. And then another question that we got from Romo Ortega Vilazian. Thank you very much for the presentation. Sorry, I just couldn't join the last part. I just only could join the last part regarding IL-6 detection. In which species was it detected? Okay, this question it was just answered. Marine or freshwater, and on which kind of sensor did you use?
So we use so for these sensors is the flexible sensors. So we always use freshwater at the moment. But one point regarding the IL-6 is IL-6 was not tested yet in any type of tanks because we had some issues with the antibodies made from fish, so we are still in the process to go to the further testing in the next months.
Thank you so much, Dr. Teixeira. The next one we got from Happiness. Does the patch have impact on health? I mean, can a human consume the fish after the analysis?
Yes, it's no harm for the fish, as is no harm for humans. Everything is biocompatible. Nothing is in contact with the inside body, and if goes to the body, everything is biocompatible and tested. So nothing affects the human to eat the fish after. Yes.
Thank you very much. I would like to say that this is the last reminder to send any questions that you might be having for Dr. Teixeira. Please write down your questions and let's start the discussion. Dr. Teixeira, I have another question for you. Considering that the fish experience stress during handling, do you wait for a certain period before you start with the recording?
So usually we should wait at least, not at least a few minutes to make sure that is stabilized, because when they actually are in transport, that can affect. So I should wait around 5 to 10 minutes and you can start, but you can start immediately. It's just you're gonna see the shifts in the sensor, and after you see the stabilization, if this makes sense.
Thank you so much. Is there any other key messages that you would like to share with us until we wait for maybe one more question to come?
We have a question that is actually very interesting from the audience. That is from Ajit Kumar. If these sensors useful for shrimp culture. So we never tried, but that is in our pipeline with the Ignition project in one of the partners. So yeah, that is something that I would like to do.
Thank you. I can see some people are raising hands. Unfortunately, we do not have the option to turn the video and the microphone, so feel free to actually type the questions that you might be having. Unfortunately, raising hands will not help us now. The next one we got is from Abdeglani Gaham. For real world application, if their fish of interest is eaten by another fish, how would you react in this case, and how would it affect the analysis?
Well, we will lose the signal for sure. if the sensor goes into another animal, so if it's not in the surface, we will lose the signal from that. How that will affect, so at the moment we are actually moving to have everything in bioplastics that could not affecting the environment, and we are moving as well to resolvable. So maybe that will be a way to not affecting the animals, but we will lose the signal for sure.
And can you reuse the sensors once they're used?
If they are, depending on the sensor, so if they are made with antibodies, no. If they are chemical, yes.
The next one we got is from Rasidian Gassim. I also lost the beginning of the presentation. The patch measures IL-6 in water or in samples, or if you can explain how IL-6 or other targets are targeted if measurements.
So as I already explained before, so these ones here, the IL-6 was just done in a lab environment, of course, mimicking water that we could have with IL-6 interference or solution. So we never did in samples because of the problem of having this antibody from fish. How can we target other measurements? So I'm not understanding what means our other target if measurement. So we can have in the same patch different cortisol, different cytokines, not just IL-6, but IL-6, IL-10, IL-8. If they are of interesting for you, we can have them in there, and the measurement happen in simultaneous with the electronics on the background. Not sure if that replies to the question, because I'm not sure what that means, but if not, you can ask again.
Or she or he can email you.
Yes, yeah, they have my contact. Yes, through Anna.
The next one we got is from Pablo. The connection between the sensors and the PC is by wireless or Bluetooth. There is any interface?
So we have by both, but from water usually, or from under something, we are using Bluetooth. We had loads of interference, so when we actually started, and we understand that even the body of the animal or the person or whatever can interfere, but at the moment we are working in overtake all of that interference, having different types of, so that is not from my side, it's from the electronic side, but yes, we are working on that.
Thank you very much. Okay, so Rasidian Gassim said, thanks. I will ask my question more specifically via email.
Thank you.
I would like to say that this unfortunately brings us to the end of today's EAStalk webinar. On behalf of the European Aquaculture Society, I would really sincerely like to thank you, Dr. Teixeira, for sharing your innovative research and insights with us. A big thank you as well to everyone that registered and joined us today and to join in contributing today's discussion. And looking forward to seeing you again on the next EAStalk webinar.
Thank you so much, Anna, and thank you so much, everyone, for your attention and for being there. And yeah, if you need anything, just drop me an email. Thank you.
Thank you. Bye.
Bye.
Fortunately brings us to the end of today's EAStalk webinar. On behalf of the European Aquaculture Society, I would really sincerely like to thank you, Dr. Teixeira, for sharing your innovative research and insights with us. A big thank you as well to everyone that registered and joined us today and to join in contributing to today's discussion. And looking forward to seeing you again on the next EAStalk webinar.
Thank you so much, Anna, and thank you so much, everyone, for your attention and for being there. And yeah, if you need anything, just drop me an email. Thank you.
Thank you. Bye.
Bye.